over, and we may need to do some exercise because this room seems to be very underheated. But let us get started. So it's my pleasure to introduce today's speaker, Drew Pardole from Johns Hopkins. And today's lecture is a special named lecture, the Charles Segal Memorial Lecture, that was established in 1989 by Charles and his wife, Edith Segal. And they were longtime supporters of the Jimmy Fund and Dana-Farber and other charitable organizations in Massachusetts and Florida till their passing. And their only desire for this lecture is that to continue to educate the future generation of medical professionals, and then those who attend the lecture take notes and study hard. So we're gonna have a test at the end of the lecture to honor the wishes of the donors. And I could not have a better speaker for this special lecture than Drew Pardole, who's a professor of oncology at Johns Hopkins, also professor of medicine, molecular biology, and pathology. So I was joking with him that looks like at Hopkins you can be a professor in every department, contrary to Harvard. He's also director of the Bloomberg Kimmel Institute for Cancer Immunotherapy at Johns Hopkins. And Drew is one of the few people who did all his training at Hopkins, starting from undergrad, graduate, MD, PhD training, and then he stayed all his career at Hopkins. And he was a graduate student with Bert Wogelstein, who I was a postdoc, and when I was a postdoc in Bert's lab, this was in the 90s, late 90s, which is kind of the sequencing era of cancer biology. Bert was always joking about Drew that, he's just wasting his time on trying to study immunology when it's, obviously not working. And, but, give a credit to Bert, Drew actually managed to turn him into the immunologist because of the convincing data and the numerous publications that Drew published in the last years. And, he's as one of the most well-known and influential immunologists in the field. The number of citations he has is over 100,000. So he has really a high impact on the field, and this has been recognized by the numerous awards that he got. He was elected to the National Academy of Medicine and also National Academy of Inventors. And today, Drew will tell us a story about the tumor microenvironment and how to convert predator to prey. So Drew, thanks for coming, and this is the plaque. Keep it here. All right.
Thanks. So I'm going to try to actually get through three components of the story. It's always both intimidating and exciting for me to come to Dana-Farber and Harvard because there are so many phenomenal scientists and also so many really good friends. I was actually hoping for the first time in 4 years to get a real snowstorm, since in Baltimore we've had nothing for all these many, many years, but I guess it sort of petered out a little bit. So the tumor we now appreciate is an ecosystem. There are a huge number of cell types that make up the tumor, and it's interesting in this particular picture, the tumor cell is just this one cell here, but when tumors grow and become clinically relevant, even though they sometimes represent less than 10% of the cells in the actual tumor mass, the cancer cell itself is arguably at the sort of top of the food chain and really subverts all of the immune cells in the microenvironment to do its bidding. One of the certainly very exciting opportunities that I've had being in this field was to be involved with some of the science and the development of anti-PD-1 antibodies, which in some tumors really does convert the predator tumor cell into prey. There are now roughly 20 cancer types in which an antibody blocking the PD-1 pathway has been approved. This is really pretty phenomenal, but I think if you just sort of look through this list, in the parentheses are the response rates. And as you can see, in virtually all of the cases, the response rates are below 50%, and even in some cases as low as 15%. So this has been certainly a significant advance, but many of us in the field now are really digging in to try to understand what determines resistance versus susceptibility to at least this one particular checkpoint inhibitor. And this area is really proliferating, and you look at papers on various aspects, and it sort of reminds me of the notion of different people looking at different parts of the elephant. Is it M2 max? Is it myeloid-derived suppressor cells? Is it Treg cells? Loss of class 1, T cell exhaustion? Is it the cancer-associated fibroblasts? And are there tumor-intrinsic elements to the genetics, the epigenetics, metabolic competition? And I think the answer is that it's probably a sum of all of these, but we have to still really understand which of these in particular cancer types are most important, so that we can effectively, in sort of a precision immunotherapy fashion, intervene in the right patients with the right therapies. We've found that one of the clinical formats that has really been a gold mine for understanding immunotherapy sensitivity versus resistance and mechanisms therefrom is the neoadjuvant platform. So patients with operable disease, in that they don't have radiographically defined systemic metastases, but you know that based on the staging of their primary tumor, there's a reasonable probability that they do, in fact, have those. They're operated on anyway, and depending, again, on the stage and the tumor type, a significant portion ultimately relapse with systemic disease. So the idea is, if you intervene with immunotherapy prior to surgery, the original neoadjuvant therapy concept was with chemotherapy was to just shrink the tumor to make it more resectable technically. But here was an opportunity to potentially use the primary tumor in place as in a sense, to enhance responses to tumor antigens systemically. And so the very first neoadjuvant anti-PD-1 clinical trial was led by Patrick Ford at Hopkins. It was a Stand Up to Cancer trial that actually literally at this point was started slightly over a decade ago, done in collaboration with Jamie Chaft and Matt Hellman and folks on our Stand Up to Cancer team at Sloan Kettering. And quite impressively, in this particular case, out of 20 patients, 9 of those patients had a major pathologic response, which in lung cancer is defined as less than 10% of the viable tumor cell, or the viable cells remaining at the time of resection were tumor cells. It turns out that the response that you're looking at in the primary tumor probably is also reflective of the systemic response. So again, you have a group of patients that pathologically either are clearly dramatic responders to the immunotherapy versus those that are not. And importantly, and this was from work recently published by our colleague Janice Taub and Julie Stein, the level of pathologic response that you see in the primary tumor correlates very, very closely with the relapse-free survival, which for an oncologist, that's what actually counts, is that you're doing something that will result in the tumor not relapsing. To us, that means that we've generated a systemic anti-tumor immune response, or at least that's the presumption. So you can see the difference between in relapse-free survival in patients who had in this particular case, it was the combination of nivolumab, anti-PD-1, and chemotherapy, a pathologic CR versus patients that did not. So that also suggests to us that by studying what's going on in the primary tumor, that's going to teach us about what's potentially relevant systemically. A lot of work in elegant mouse models by folks like Max Krummel, some of what we saw by looking at tumor-draining lymph nodes in the clinical trial, which I won't show you the data on, but certainly supported the notion that not only is PD-1 pathway blockade working within the tumor, but in a setting like this, its function in enhancing T-cell priming in the tumor-draining lymph node by dendritic cells that picked up tumor antigens in the tumor and trafficked to the draining lymph node, resulting ultimately in enhanced T-cell activation going through the conventional pathway through the efferent lymphatics, getting into the bloodstream, and then eventually leaving the bloodstream and going and seeking out micrometastatic disease is, we think, a reasonable probability. So back to the elephant, I'm going to tell you sort of about 3 components to the elephant. The first one is it T-cell exhaustion? We are reluctant to use the term T-cell exhaustion because having been raised as an immunologist, that's something that was a term applied to chronic viral infection. So I think that we often use the term T-cell dysfunction as sort of a broader term. But the first 2 stories are going to be in the context of neoadjuvant immunotherapy for lung cancer. This was a lot of great work done by some wonderful folks. These are some of the key folks involved, and I want to specifically call out Kelly Smith, who started this work as a postdoc with me. She's now a famous cancer immunologist and is now an associate professor already, and she's been kind enough to allow me to continue to work with her on some of these really cool projects. So the first cell type that we went after, as do a lot of people in this field, were CD8 cells. And one of the beauties of the neoadjuvant platform is that when the surgeon resects the tumor, you just get a ton of tissue. So you can study a lot of cells. And so ultimately the approach, and this was, a lot of this was the work of Justina Cauchy, a graduate student that worked with Kelly and myself, was to combine two technologies. One that Kelly developed called the Manifest assay on peripheral blood, which stands for mutation-associated neoantigen functional expansion of specific T cells. But everybody can remember Manifest, which is essentially a way using TCR-seq on T cells from the peripheral blood expanded in culture with peptides derived from mutations in that individual's tumor. So you define T cell candidate T cell receptors from neoantigen-specific cells, and then you do this combined single-cell RNA-seq, TCR-seq on tumor-infiltrating lymphocytes, so you can actually look for the T cells in TIL that are candidates to be neoantigen specific based on the Manifest assay, and we actually can validate this by cloning out the T cell receptors and putting them into a JERCAT system. This is all the 10x system. I finally learned that why they call the company 10x is because they charge 10x what's in your budget. So what you can then do, and I'm going to assume, because this is actually the most sophisticated audience that I've ever given a talk to since the last time I spoke at Dana-Farber, that you all pretty much know the basics of single-cell transcriptional analysis as well as TCR-seq. It's really a transformative technique. The idea is that you can pick out the small proportion. In lung cancer, it really looks like it's probably 1 or a few percent in the ocean of TIL that are tumor specific. And again, we define these as manus specific, again, because we can, with whole exome sequencing, define the mutations. And there are a number of things that you can do with this. You can discover genes that are associated with dysfunction versus reactivation after checkpoint blockade, in particular, linking those to the pathologic response that one sees. And also one, as it turns out, can clone out T cell receptors specific for oncogenic driver mutations. And these actually have translational significance because these studies can at least nominate new targets based on their differential expression patterns in responders versus non-responders. And actually, I'll tell you a little bit about the generation of antibodies against shared oncogenic mutations that was actually inspired by this work. So because we're looking for a very small proportion of the total TIL that are tumor specific, what it meant was we needed to single cell a large number of cells. And this is where I'm really thankful to my budget administrators who somehow managed to protect me from them closing down my lab. And so this is the UMAP from nearly 250,000 CD8 cells from that original neoadjuvant PD-1 lung cancer clinical trial. And what you see here are a number of clusters that we designated tissue-resident memory cells. Some effector cell populations here, stem-like memory cells, there's a mate population, a whole bunch of different populations. And for any of you who've done single cell, you know that what you designate a cluster, oftentimes there's sort of no Bible that tells you that you have to call it this, and get into that in a moment. The reason we call these tissue-resident memory is because these populations have very high levels of two genes that are fairly canonical for tissue-resident memory cells, the integrin CD103, and also the transcription factor Hobbit, which is also called ZNF 683. So I mentioned to you that we could define, I'm going to call them mano or neoantigen specific cells, but when we do the manifest assay, we also use a set of EBV, flu, CMV, sort of as control peptides. So that also actually gives us T cell receptors from T cells that recognize these viruses. And indeed, you can find in tumors not only T cells specific for the neoantigens, but also specific for various viruses. And so this is looking at manner-specific T cells, the red triangles, EBV-specific T cells, purple, and flu-specific in blue. And what you can see quite strikingly is they map to different places. The majority of the manner-specific T cells, these are CD8 cells, map to a few of the tissue-resident memory cells in the population, with a few spilling over into this one effector population, which is actually where you see the majority of the EBV cells. Virtually all of the EBV-specific cells are essentially active effector T cells, probably because when we see them, it's somebody who's recently reactivated their EBV, and so their EBV-specific T cells are getting a boost. The flu, we found a lot of flu-specific cells. And if you think of it, influenza is a respiratory virus. And indeed, now, if you look at various and sundry other single-cell data sets, the cancer type with the largest number of flu-specific cells is not surprisingly lung cancer. These flu-specific cells, which are classic tissue-resident memory cells, are in a distinct TRM population from the tumor neoantigen-specific cells. So it's certainly a very interesting pattern, and it shows you the distinct transcriptional programs that tumor-specific cells have. You can find overlaps between genes upregulated in the manner-specific cells and EBV. You find that's actually the major overlap that you see, and in fact, they have the transcriptional programs that are clearly of a cell that is recognizing antigen and trying to be activated and trying to eliminate its antigen. A couple interesting things that you find that are unique to manner-specific cells. One is actually CD39, which is a checkpoint involved in adenosine production. The chemokine CXCL13, we have absolutely no idea why that's selectively expressed in manner-specific cells. They tend to have, in lung cancer, very low TCF7, which is a stem T cell gene, and they also have quite low levels of the IL-7 receptor, and I'll get into that in a moment in terms of responders versus non-responders, actually here. So the manner-specific cells do have high levels of the standard checkpoints, PD-1, CTLA-4, LAG-3, TIM-3, TIGIT, CD39. And without question, the non-responders have higher levels of checkpoints than the responders. And this does certainly argue, and it generally is across the board, some more than others, but it certainly is an argument for checkpoint combinations of checkpoint blockers. But also, interestingly, even though IL-7 receptor levels are relatively low on neoantigen-specific T cells, they're quite a bit lower on non-responders than they are on responders. And this has actually motivated a clinical trial that we're trying to broker between actually a local company that has a half-life extended IL-7 together with anti-PD-1. But also, if you look specifically at the manner-specific cells, at genes that are differentially expressed comparing the responders versus the non-responders, which is a very, again, clinically relevant and biologically relevant dichotomization. Some of the most interesting things are genes that are upregulated in the non-responders, a number of which are membrane receptors that are certainly amenable to intervention, such as Lalin, a number of the killer inhibitory receptors, which were defined originally on NK cells, but also can be expressed on T cells. The other thing that we found, and this is maybe a little bit scary, but is that when we actually cloned out the T cell receptors, again, you put them into a JERCAT reporter line, and then you look at ligand-dependent signaling, which is essentially you do a dose-response curve with increasing amounts of the cognate peptide and look at the response. What we find is that there is a huge shift such that you basically need much less peptide to stimulate clones from major pathologic responders, the responders that have a ligand-dependent signaling quality that's really very similar to flu and EBV-specific T cell receptor pairs, much less so, about an 80-fold weaker response, quite specific but nonetheless weaker in the non-responders. What this tells us is that at least what we can grow out has a lower quality T cell receptor, even though they're nonetheless specific for tumor antigens. And it's something that we certainly need to contend with. While we were doing this work, actually, we found out that Kathy Wu's group, Kathy, good friend for years, was actually doing a very similar analysis in melanoma, and with the magical hands of Giacomo Oliveira, were defining initially melanoma-specific CD8 cells. And I have to say, I was always very jealous because in melanoma, about a quarter to a third the TIL are actually tumor-specific, either neoantigen specific or TAA specific. So actually recently, Jaja integrated the data. So this is actually data from Giacomo's paper. And essentially the red here are mana-specific T cells. The purple are tumor-associated antigen like MART1 melan-A specific T cells, and then viral-specific T cells, which included things like CMV and all. Far fewer flu-specific cells in melanoma, because flu is not a cutaneous virus. In also an effector memory population, and they annotated their some of these clusters as actually exhausted T cell clusters, which is where most of the mana-specific cells are, although some clearly in an activated cluster. So Jaja actually merged our lung cancer and their melanoma data into one UMAP, and then you split them out. And so this is actually using our nomenclature. But if you go down and compare the melanoma and the lung cancer, what you find is that they're actually strikingly similar. Most of the mana-specific cells, as well as the tumor-associated antigen-specific cells in melanoma, are in these populations, which are which are tissue-resident memory cells, but they also have features of exhaustion, largely high checkpoint expression, tox, and whatnot. But what the melanoma TIL have, to a much larger extent than lung cancer, are a population of cells that are in this T effector population, which may go along with the fact that, for reasons that we still don't totally understand, melanoma is much more immunogenic than lung cancer. And most of the patterns of checkpoint expression were actually relatively similar. Here, again, showing you that CD39, the gene name is NTPD1, is particularly high on mana-specific cells, both in melanoma and in lung cancer, also in the TAA-specific cells, but not in any of the virus-specific cells. And then in looking at activation markers, interestingly, one of the big differences that stood out, which goes along with a larger number of effector T cells in the melanoma, is this very important granzyme K, which is found in an early effector type T cell, which was virtually absent in the lung cancer mana-specific cells, but with a significant population in addition to negative, so these two populations in melanoma. So just to, again, give you a sense of the complexity of what you see, and disease-specific similarities and differences depending on the type of cancer that you're looking at. I also mentioned this notion of potentially targeting shared oncogenic mutations or mana. And in fact, using this T cell receptor transfer system, even not specifically looking for T cells that could recognize oncogenic mutations, we nonetheless found them. In fact, particularly, we found quite a few that recognize various shared mutations in p53. This is an example of 4 clones in a particular patient that recognize the second most common p53 mutation, R248L. And this actually led to now, it's I think an 8-year collaboration to actually develop therapeutics targeted toward mutant oncogenes. Some great students involved, Jackie, Michael, Brian, Emily, Alex, mentored by handsome, youthful cancer immunologist and cancer geneticist. So the idea is that you can make high-affinity antibodies if you have really amazingly brilliant, talented students that act like a T-cell receptor but have higher affinity than T-cell receptors that are peptide MHC selective. And it turns out that there's pretty reasonable presentation of the most common p53 mutation, R175H, on the most common HLA allele, which is HLA-A2. So this has to be specific for both the peptide and the HLA. And then you link that to an anti-CD3 antibody. So it's totally tumor specific. And you can see this is just an in vitro assay of various cell lines. And the bottom line is that there is essentially no induction of T cells unless the tumor is both HLA-A2 positive and has the R175H mutation. And you can actually, Sandra Gabelli's group actually did the trimolecular crystal structure of this particular antibody, recognizing the p53 HLA-A2. I should say that this actually docks on peptide MHC, this antibody, in a fashion that is very different than conventional T-cell receptor docking, but nonetheless retains specificity and also activity. These are just 2 different concentrations of the mannabody CD3 bispecific in mice bearing a tumor. This is the isotype control, and you can see that there is a healthy anti-tumor response. This despite the fact that there are a very small number of peptide MHC complexes on the surface of the cell, but what we now actually know is that the number of these complexes are probably increased because gamma interferon production by T cells is very sensitive with these bispecifics. That gamma interferon drives upregulation of HLA, and so there you get a higher density of peptide HLA. Okay, next part of the elephant, Tregs. So because we had a single cell, so many cells to be able to identify the manna-specific T cells, we also had a lot of Tregs. In fact, because you do typically see a much larger proportion of T cells that are Tregs in tumors than, for example, the peripheral blood or in normal tissues, we actually had within our single cell library almost 75,000 Tregs, and we were able to actually cluster these into 10 or 11 different clusters. One of the clusters that stood out of interest was this one cluster here, and this is a heat map that that were particularly high in expression of two TNF receptor superfamily members. One is called OX40 and the other is called GITR. These also tend to have a lot of LAG3, which is another checkpoint that we actually worked on from its inception with Dario Vignali starting about 25 years ago. It's now actually approved in combination with anti-PD-1 in melanoma. And in fact, if one separated out pathologic responders from pathologic non-responders, and we also included some single-cell data from some untreated patients, what we found was that there were a larger proportion, significantly larger proportion of these cells in non-responders than in responders. And if you just do a volcano plot for differential gene expression using a pseudobulk program, you actually see a lot of genes that are selectively expressed in this OX40 high, GITR high population. You can see these are way more highly expressed, but also a number of other interesting targetable molecules, including IL-2 receptor alpha, so that's CD25, which is, and also CCR8, a chemokine receptor. And so these actually, and in fact, now together with BMS, CCR8 antibody is actually just coming into clinical development. And again, in TIL Treg, in the lung cancers, again, if one actually looks at expression of cell numbers that express these genes at high levels, they are much higher in the non-responders than in the responders. And one can actually sort out the OX40 high, GITR high, LAG3 high Tregs from the rest of the Tregs and perform an in vitro T cell suppression assay. And roughly 90% of the suppressive activity in TIL Treg come from this one population of cells, which on average is about only 18% of the total FOXP3 positive Treg. I'm going to actually skip this in the interest of time. So Arbor Dikema, who was the student working on this, spent a lot of time trying to identify tumor neoantigen-specific Treg cells, encouraged in part by a beautiful paper that Giacomo published on these huge clones of CD4, both T conventional and Treg cells that indeed recognized mutations in their melanomas. And as hard as she tried, Arbor was unable to find any. And in fact, when we looked at the clonality, we realized that this is a case where lung cancer and melanoma were really very, very different. We could barely find any Treg cells that had more than 5 or 6 cells of one clone, and most were just a single cell, which was just completely different than in the melanoma. So just completely different biology. So to try to get a sense for these, what Arbor did was go to a mouse model where, without going into the details, essentially you take T cell receptor transgenic Tregs from a mouse, and these are central Tregs, and you transfer them into a mouse bearing a tumor that expresses the cognate antigen, and you see what they turn into. Now, I can't tell you how many thousand times I've been asked the question, what good is a mouse model in teaching us about the human? And one of the things I say is that you have to understand, it helps to have validation that what you're looking at in the mouse looks something like the human. So for example, it turns out that murine T cells don't express CXCL13. So that CXCL13 that's really interesting in human TIL that are tumor specific, we can't use the mouse to study that because this is just something that the mouse is just different from the human and doesn't model. So we were happily surprised to see when we single-celled the Tregs from the murine tumors that we found populations that were really very analogous to the human. And in fact, this is just showing you that we can find an activated Treg population that looks very much like this activated GITR high, OX40 high population with LAG3, CD25, EBI3. There's this interesting population with an type 1 interferon signature. We have no idea what that is, but that looks very similar to a population between mouse and human. There's a resting population that looks very similar that has CCR7 cell, the S1P receptor, and also this other interesting population that we saw, again, both in the mouse and in the human, that really looks kind of like a Th1 cell, so that has reasonable levels of gamma interferon, various Th1 chemokines, and it also has various granzymes that you typically think of in cytolytic T cells. So the question then is, where do the tumor antigen-specific Tregs, and again, we've sort of rigged this system, so we're specifically looking at Tregs that we know have a T cell receptor that recognizes an antigen that's only in the tumor. And the huge surprise was that virtually all of them differentiated essentially into this Th1-like population. The control being the same tumor, but without the antigen. And there are very few of the antigen of these GP-specific Tregs that you see, but the few that you do see are actually in this resting population. So that is what we would expect because their cognate antigen is not in the tumor. So if you now look at these cells, and actually Arbor was able to look at the differentiation of the original transferred naive T cells at different time points after they entered the tumor, what you saw was this increasing set of genes. This is actually a gene score that are Th1-like and cytolytic, while FOXP3 started decreasing. And this was reminiscent of this phenomenon first described in autoimmune disease by Jeff Bluestone, which is somewhat controversial, but I think it's absolutely real, of the so-called X-Treg. Namely that in certain environments, Treg cells are fragile. They can lose FOXP3 and they can actually acquire a conventional CD4 T effector cell phenotype. And along with this Th1 looking pattern, indeed, they upregulate Tbet, which is the canonical Th1 transcription factor. Everybody in this room should know about this because your boss actually discovered that. And we went to a lot of lengths that I won't go through to show that that these are not just transient T conventional cells, but this is really a differentiation of Treg cells. And in fact, what we found is that there are a set of genes that generated a score that we could find this population now in the human TIL Treg. And lo and behold, we found the exact opposite is what we saw with the suppressive OX40 high, GITR high Tregs, which is that now they are much more prevalent in the responders than in the non-responders. And in fact, if we compare now non-responders versus responders and do a trajectory RNA velocity analysis, which is sort of looking at a snapshot to give you pathways of differentiation, we find that they're in the opposite direction. In responders, basically cells are differentiating very actively toward this Th1 type population, whereas in the non-responders, they're actually differentiating out of the small population that's still there and actually into this activated suppressive population. So obviously the key question that we want to answer, we have some hypotheses that we are testing, is a question of what actually forces a Treg in the tumor microenvironment, what forces drive it to go into the suppressive pathway versus a so-called X-Treg, Th1-like pathway, which should be enhancing anti-tumor immunity. And there are a couple approaches that we're using to follow that. So I want to just wrap up. I know it's a little bit late, but this is actually the coolest story of all, which is an MDSC story. It's not a neoadjuvant story, and it has to do with glioblastoma. This is the great work of a wonderful neurosurgery fellow, Christina Jackson, who's actually now on the Penn faculty. And so if you look in glioblastomas, you find very few T cells. So these are, I think in this whole field, these yellows are T cells, probably about 5 or 6 cells. Now, you can, if you're really good, and actually, Kathy actually published a beautiful paper showing that you can find an occasional T cell that actually recognizes a neoantigen in GBM. So it's not zero, but they're few and far between. Other than the tumor cell, the major immune cell that you see in GBMs are actually myeloid cells. And so Christina focused on myeloid cells in GBMs. And there are a lot of, and again, this is a lot of single cells. One finds multiple clusters of microglia. These are these MCGs, various populations of macrophages, and also a number of MDSCs, including one that looks like a MDSC progenitor, which was described by Dmitry Gabrilovich, and early MDSC or EMDSC, MDSC, granulocytic MDSC. So a lot of these cells. And what Christina was interested in was which ones are only selectively found in the stage 4 GBMs. It turned out that the two major populations that are exclusively found in grade 4 GBMs are these two myeloid-derived suppressor cell populations, these EMDSCs and MDSCs. In grade 2 and grade 3, they're virtually non-existent. So profiling them, one thing that you find is that they are unbelievably metabolically active. We sometimes think of MDSCs as being very quiescent and not metabolically active. These are incredibly active both for anabolic and catabolic pathways. Jonathan Powell's group had developed this antibody-based so-called metaboflow with antibodies against various enzymes involved in various metabolic pathways. These are the two MDSC populations in GBM, and you can see they basically light up for enzymes involved in glycolysis, oxidative phosphorylation, fatty acid phosphorylation, the mTOR pathway. They're highly active. These cells have regulons regulated by a number of key transcription factors that activate metabolic programs, and they also have regulons that are activated by the hypoxia pathway, suggesting that they're living in hypoxic conditions. She also was able to profile the tumor cells. This has been by single cell, and found that there was, interestingly, one population called T4, whose representation in the tumor cells correlated almost perfectly with these early MDSCs. And it turns out that this population is a glioma stem cell. It's got all of HES1, KLF4, GLE3, basically all of the genes associated with cancer stemness. Again, not to get into the controversy about what cancer stem cells are, but clearly this has the features of a cancer stem cell. And again, its representation correlates with the early MDSCs. And if one does a cluster-cluster ligand receptor interaction map using a program like CellChat, what you find are some interesting relationships between the eMDSCs and this T4 glioma stem cell population. In particular, there are a number of chemokines that the T4 population is making that link to receptors on the eMDSCs, as well as a number of growth factors being produced by the eMDSCs that link to receptors on the T4 population, in particular FGF11 made by the eMDSCs, which is a ligand for FGFR1, which is expressed at high levels on the T4, and also this midkine that is a ligand for EGFR. And in fact, if you correlate the expression levels, for example, here of FGFR1 with essentially the regulon score, so the genes, expression levels of the genes that FGFR1 is known to regulate, you do see a correlation coefficient with a very highly significant p-value. Similarly for EGFR and the EGFR signaling score. Now, does this all link together, or is this just sort of going down a computational rabbit hole? Well, it turns out, interestingly, that the major genetic subset that you see in terms of mutations for gliomas is actually a mutation in IDH1, which is a metabolic enzyme that normally produces alpha-ketoglutarate, but the mutants produce this so-called oncometabolite, which is 2-hydroxyglutarate, which turns out to actually inhibit TET2 which is essentially a DNA demethylase. And it turns out that the very chemokines that these T4 glioma cells are producing that attract the EMDSCs are in fact hypermethylated in the IDH1 knockouts, and that actually correlates with a much lower expression of all of these in the IDH1 mutants. In fact, if you now separate out your gliomas into IDH1 mutant versus IDH1 wild type, it turns out that the IDH mutant gliomas actually don't have any EMDSCs in them. Only all of the EMDSCs that you see, similarly with the MDSCs, are actually in the wild type and not the mutant. So what was sort of emerging was this notion that maybe you have these EMDSCs that are talking to this glioma stem cell population, and that's driving badness in the tumor. It had been shown that tumors that have a lot of the signatures of these glioma stem cells were essentially bad actors. Those patients did worse. In working with some of the pathologists, they reminded me of something that I learned in pathology when I was in medical school, that was pre-World War II, called a pseudopalisading region. So this is a pathologic feature that is pathognomonic for bad acting gliomas, and this is what they look like, and they sort of look like a palisades, and generally around an area of necrosis, and it's a very distinct pathologic feature. So with the help of Vasan Yegna-Subaranian and C.J. Lucas, who's the pathologist, and Ruan Wang, we actually delved into the land of spatial transcriptomics. And so this is a Visium analysis, which is actually very cool. And so this is, these are essentially these spots that are not a single cell, but they're a few cells that you overlay on the H&E. And so this is actually an example of an H&E section of the glioma. This is a necrotic area. This is an area full of tumor. And then this is actually the pseudopalisading region. And so this is what the spots overlay looks like. And the analysis is done such that each spot is the equivalent of a transcriptome package. It's not a single cell, but it's what you have in the spot. And the program, in a very similar way, can either generate in an unsupervised way clusters. And so you can see that this is one cluster here, there's another cluster here, there were 9 clusters that were generated. Or you can actually ask it sort of in a feature map way to say, well, where do you find signatures of a particular cell, like an EMDSC or a T4 tumor cell? And the results are actually quite stunning. So just as a marker are these white dots here. And so this is actually the EMDSC signature superimposed. And you can see that right along the pseudopalisading region are the EMDSCs. Right next to and overlapping the EMDSCs is the spatial signature for the T4 glioma stem cells. And then interestingly, T3 was a population that looked like from pseudotime, it had on the single cell, it had differentiated from T4, and lo and behold, this is basically the T3s that are essentially adjacent to the T4s. So there, and in fact, one can actually do these pseudotime analyses and actually demonstrate this spatial pseudo-differentiation going on. You can then start looking at specific genes of interest, and one of the genes that stood out, which of great interest because there's an antibody against it, is CXCL8, which is also IL-8. BMS has an anti-IL-8 antibody, so now we're actually trying to get them to repurpose this potentially for GBM. You also can see, it's not quite so easy to see, but you can see some FGF11 in the pseudopalisading region and also FGFR1. The FGF11, my computer just ran out of juice, which was great because I just come to the end. So that's one way to make sure you don't go too far over. So that is the third scenario. And my last slide was basically going to go back to the elephant and to say, where do we go with this? And I think the answer is that we have technologies and we have clinical settings that we can apply them to, which hopefully link to the appropriate studies that one can do in the mouse as well as in vitro systems to really begin to define the biology of these various players and to link them to response and to clinical outcome. I think what we now need to do is to integrate these and remember that these are all working in an ecosystem and to determine which of these factors in given tumors are the most important players in terms of this predator-prey interaction. I think that's something that we're going to have great opportunities over the next 20 years, and I'm glad to see a lot of young faces in the room because you all really do have a cornucopia of opportunities in front of you now, and a lot more to be done to translate those into benefit for patients. I'll end here. Apologies for going a little bit over, but... And thank you to my computer for keeping me on time.